Thanks so much for inviting me today to talk about biological impacts of climate change. It's a big topic. There are a lot of folks at Ohio State that work on the linkages between biology and climate change. This is the list that I could come up with in one afternoon, but there are more, so I apologize if you're not on this list, but you should be. And you can see from this list that we span 4 different units and range in applications or investigations from the subcellular all the way to the ecosystem scale. I study coral, and so I'm going to use this as a model example of how climate change affects biological systems, but you could tell this story for forests, for savannas, for pick your ecosystem or pick your species of choice. The story is similar. So coral reefs are these amazing ecosystems, but they are under threat. Over the previous century, the oceans, tropical oceans warmed by almost 1 degree Celsius and became 30% more acidic. Over the current century, if we continue emitting CO2 at the rate that we are, according to IPCC, the tropical oceans will warm by 1 to 3 degrees Celsius more and become up to 150% more acidic. As CO2 increases in the atmosphere, not only is it a greenhouse gas that warms the atmosphere and the ocean, but about 1/3 of that, 25 to 30% of that CO2 gets absorbed into the surface ocean, reacts chemically with the seawater chemistry and makes the water more acidic, and that's called ocean acidification. Warming is bad for coral. It causes them to bleach and die. Ocean acidification is bad for coral because it interferes with calcification and their ability to compete for space. The two together are horrible. So coral are these amazing organisms. I've zoomed into a coral here, and it's made up of these polyps. The polyps are, do I have this? Perfect. The polyps have tentacles, and they use that to capture zooplankton that they stuff into their mouths and eat. And that's the way they get nutrition, nitrogen, phosphorus, and other nutrients that are essential for biological life. But they're also symbiotic with single-celled algae, and those algae reside inside the animal's cells. And they are plants. They come in densities of up to 1 million, or actually normal concentrations are 1 million per centimeter squared. That's about the area the size of the end of my finger. That's the concentration inside the coral animal. And they photosynthesize far in excess of their own needs, translocate the majority of that photosynthetic sugar to the coral host, supplying it with fuel. Sugar is just raw fuel. And in exchange, the host can share nutrients that it gets from eating zooplankton with the algae. So this is a very tightly coupled symbiosis, and it works really well. But the symbiosis is threatened by temperature. So I'm showing here a picture of a healthy coral reef with an F. And corals are normally greenish, yellowish, brownish in color because of the algal cells in their tissue. So this is a normal looking healthy reef. But when temperatures increase by as little as 1 degree Celsius for as little as 10 days, that's 1 degree above normal summertime maximum temperatures. It causes stress and a dissociation of the symbiosis. The coral eject their endosymbiotic algae, and you see through the clear gelatinous tissue of the animal to the white skeleton underneath, hence the term bleaching. The coral aren't dead, but they're highly stressed, and without that photosynthetic sugar, they are starved. If bleaching conditions last for too long, they start becoming more susceptible to disease and mortality. Bleaching is now occurring in back-to-back years. This was not expected to occur until 2030. We are now ahead of schedule of when we thought we would see this. And for example, in 2015, 2016, there were 2 back-to-back bleaching events on the Great Barrier Reef in Australia. And in that time, 60% of the corals in the northern half of the Great Barrier Reef died. This is equivalent to 60% of all the forests from Maine to Georgia dying in a 1-year period. So these are large catastrophic events that wipe out the foundational species that form reefs. And without them, reef ecosystem function starts to collapse. Temperature is the single largest threat to coral reefs right now because of the effect it has on bleaching. And it is resulting in decreases in coral abundance, and we expect to see decreases in diversity or species losses, at least locally, if not globally, for some species. Ocean acidification is the other CO2 problem for coral because it interferes with calcification. When you disrupt the carbonate equilibrium in seawater, the pH drops, and as a consequence, we also have decreases in carbonate, which is essential for corals to form calcium carbonate skeletons. By mid-century, we are expecting to see net dissolution on reefs because of ocean acidification. So why should we care if reefs disappear or get worse anyway? Well, reefs provide incredible ecosystem services and economic value. They provide food for human nutrition and subsistence living, barriers that protect coastlines from storms, flooding. They supply sand to beaches, which is critical for supporting a tourist industry. Many developing countries, tourism is the single largest source of revenue to their GDP. At least 100 countries have coastlines with coral reefs, and almost 1 billion people live within 100 kilometers of reefs. It has been estimated that their economic value is $9.9 trillion globally. So losing coral reefs would have huge socioeconomic impacts. And we are losing reefs right now. They also have immense ecological value. They provide food for marine organisms, critical habitat for mating, hiding, sleeping, refuge from storms and predators for the small invertebrates and fishes. And coral reefs house 25% of all marine species, yet occupy only 0.2% of the surface area of the ocean. They are disproportionately responsible for marine biodiversity and are considered the rainforests of the ocean. So they have enormous economic value for humans, and they have critically important value ecologically for marine ecosystem function. So coral bleaching and ocean acidification are really bad for reefs. But every time there are bleaching events, there are always some that survive. There are always some that either don't bleach in the first place or recover really quickly. And I'm interested in understanding this resilience. Why are some coral resilient? What underlies that resiliency? And this is a picture I took in Hawaii. You can see these two coral are side by side. They're on the reef, exact same environment, flow, light, food, everything. One is bleached and one isn't. And why is that? So we've done some experiments to try and tackle this, because if we know what traits underlie coral resilience, we can then target that for conservation and protection purposes. We can make evidence-based decisions in how we manage coral reefs and how we protect them to buy us time until we start mitigating CO2 and reduce that global threat of temperature and ocean acidification. This is really short-term management that we're trying to optimize. So we've done some experiments, and I'm gonna introduce you to the results from 2 of them very briefly. We stressed 2 species of coral, Acropora millipora and Turbinary reniformis, with both elevated temperature and ocean acidification stressors equivalent to what we expect at the end of this century for 24 days. So we took a coral colony and we took fragments from multiple colonies of both species. Half were put under conditions mimicking today's environment, and half were put under conditions mimicking future ocean temperature and pH. And after 24 days, we harvested the corals and made a bunch of measurements. And you can see that for Acropora millipora, under today's conditions, it looks really good. Under future ocean conditions, not so good. About half of them started looking pale and sickly after just 24 days. Turbinary reniformis looked great under both conditions. So what is it that makes one look great? And is it great even if it looks great? Is it physiologically still doing well? And same for Acropora. So we made a suite of measurements, which I will not get into, but I will summarize. And we found that in Acropora, there was reduced algal health in several metrics that we measured. There was reduced host health, including skeletal dissolution. So there was net skeletal loss. And they were unable to maintain a stable microbiome. In humans, we know the microbiome is associated with healthy immune function, and we think the microbiome in coral does the same. We don't know exactly how, but we think it's associated. In this coral, the microbiome was not only was it unstable, there was a massive loss in diversity of the microbes associated with it. And the ones most responsible for that change are associated with either coral disease or plant disease. So we find that this is a negative impact. And concluded that this species is not resilient to future ocean conditions. This is probably true for most Acropora. There are about 70 Acropora species. They tend to be the first to bleach and are heavily affected by bleaching events and with high mortality rates. Turbinaria reniformis, on the other hand, was physiologically very stable, had a stable microbiome, and also had twice as much protein as Acropora, 25% more energy reserves, and a lot of mucus. Energy reserves provide buffer against bleaching stress. If you can't photosynthesize to get fuel, at least if you can get more, you have more fat to catabolize, you can survive. And they have a lot of energy. The mucus is also the substrate that houses the microbiome, and they have a lot of it, and maybe that's why their microbiome is stable. Either way, this species has all kinds of great traits that would make it resilient. High energy reserves, stable microbiome, a lot of mucus, and it also houses the endosymbiont species Durasdinium D1A, which is thermally tolerant. Altogether, this species probably can withstand quite a bit of stress, and there may be more like it. And it suggests that there is resilience out there. It may be greater than what we have predicted. We've also looked in nature for supercorals, and we have found some in the Gulf of Eilat, which is in Israel. The three species here that we studied, Stylophora, Passalopora, and Favia favis, are ubiquitous throughout the world's oceans. But in Eilat, they are special. It took 4 times the heat stress for them to bleach. It took plus 6 degrees Celsius for 37 days before they bleached, whereas most corals take a quarter of that before they bleach of heat stress. So what is it that underlies it? It's not thermally tolerant endosymbionts. That D1A doesn't exist there. But they do have extraordinarily high energy reserves. And in one case, at least, high baseline feeding. Again, another mechanism to compensate for low food supply is being able to eat more. And we've seen this in other studies, either the capacity to increase the amount of feeding on zooplankton or have high baseline feeding will compensate for that photosynthetic fuel loss, and those coral either don't bleach as much or recover more quickly. But these are the same traits that we find in all corals everywhere, just in these corals it happens a lot later with more stress. So we think there's something genetically unique about this population, and that these might be source stock for reseeding reefs that are devastated by bleaching elsewhere in the world. Now, species introductions, highly contentious, but it is on the table because desperate times call for innovative and more riskier decision making. So how will corals survive in the future? Being better feeders, having high energy reserves, stable microbiome, thermally tolerant endosymbiote types. These are traits that show up over and over in different studies that we've done. The supercorals and the ones that have multiples of these traits are the ones that we should be targeting for coral conservation, management, and restoration. We should be using this information to be making good policy decisions, evidence-based policy decision making. But we need to evaluate whether or not these traits are, in fact, ones that lead to resilience when corals are exposed to chronic stress of temperature and ocean acidification. So I encourage you to go see my students' posters, Carrie, Rowan, and Jamie. They're here, and their research is starting to look at these long-term chronic exposures and what traits are emerging when you have that. So the solutions for biological impacts of climate change, I've told a story about coral reefs, but it applies to other ecosystems as well. We need to use evidence-based we need to use evidence to base our decisions for both conservation, management, and policy in the short term so that we can preserve ecosystem function long enough to bridge the hump until we start reversing the curve, until we start bringing CO2 emissions down and reduce that thermal threat. Because without some kind of conservation effort, we might not make it to the hump reversal. But if we don't change the CO2 emission rate, it is unclear whether coral can infinitely adapt or acclimate to ever-increasing temperature and CO2 and lower pH. To get to this point, we need to have apply political pressure to change regulations and be actively involved. As scientists, we're not taught to be activists, but I feel like we need to play a role in being actively involved in sharing what we know, being willing to speak out about what we know about our science, to share that and influence decision makers and policymakers. Thank you.
Let's make time for a quick question. Yeah.
Hi, sorry if you mentioned it, but I'm just curious, can bleaching be reversed?
Well,
Real quick, the question was whether bleaching can be reversed.
when the temperatures, if temperatures go back down after a few weeks, you get almost 100% recovery. But the longer that thermal stress event lasts, the less recovery there is, because coral will eventually just repopulate their endosymbionts and get their color back. But even in a mild bleaching event, it can take a year or longer for full recovery to occur.
All right, thank you again, Andrea.